Okay. All right. Okay, perfect. So our tutorials will be recorded for students who cannot join us synchronously, and then the recording will be posted on YouTube, and all of you will receive a link to the YouTube video for you to review the video. So we'll also send out email reminders for the Saturday tutorials. Each tutorial will cover different parts of the Brain Facts book or a different part of neuroscience discipline. So, and then what's really new this year is that we're trying to build neuroscience at home toolkits. So these are little toolboxes that provide you with raw materials of demos that you can really interact and explore on your own. So at the last tutorial, that is February 12th, we'll have staff members that will guide you through the toolkit online. So it's a really hand-on experience rather than, sitting in a potentially boring lecture. So at the day of the Brain Bee, so on the 26th, we'll start with some really fun icebreakers. We'll bring college students here at WashU, and it's a good time for you to just, connect with the students and really talk about what their college experience is like. We'll also do fun and interactive demos. And then we have a trivia style written quiz and an oral competition. So it's really just like a spelling bee. And then we'll have neuroscientists and physicians want to come in and speak to you about their experience, about their career growth, about the exciting thing they're doing. And lastly, we'll also provide really great prizes. So all of these activities are free, but we do require registration. So there's a website over here that you can really just go on there and fill out a form. See, there's a so someone asked, is Brain Bee in person this year? Unfortunately, no, this is fully virtual. But the benefit of that is you can really do that from the comfort of your home. And we have students from out of the state to join us. So we're going to bring in more people. So we do have experience with moving the whole Brain Bee event online like we did last year. So we're pretty confident that we're going to provide you with a good experience. And that is well complemented with your actual exploration through the Neuroscience at Home toolkits. So again, we do ask you to register. I'll send you an email about registration. It's a really short form to fill out. Basically, we just wanted to keep you abreast on, any news about Brain B or any event that we're providing. It's the best way to stay in touch with us. Okay, and I do want to just talk about some study materials. So because there is a trivia and there is a oral competition, we recommend some of these materials. The first thing is called the Brain Facts book. This is where all of the questions will be deriving from. We'll only ask about terms and concepts shown in this book. And this book is you can download that for free at brainfacts.org. It's a really short read, but it's really, a good introduction to neuroscience. And then also on this website, there's a 3D brain model. So we'll be talking about some brain anatomy, different parts of the brain. But on this website, they provide you with this powerful tool where you can just rotate around the brain to see different parts at different angles. So traditionally, if you're in person, well, we're going to actually bring in a human brain specimen for you to really touch and see. Unfortunately, that's not really doable online. So we thought this model would supplement that. And lastly, we'll have neurons at home toolkits. So just as a preview, one of the things that's really exciting for us to do in this toolkits would be visual illusions. So we'll build visual illusion cards and really for you to trick your visual system a little bit. So that is a wonderful way to learn about how the visual system works. Okay, so are there any questions about logistics? Feel free to put them in the chat. We'll answer them, or feel free to just unmute yourself to shout out any questions. Okay. All right. So welcome officially to your first tutorial. So this tutorial is not meant to be a lecture, and we're in no way are going to provide you with a comprehensive, test prep for neuroscience. We don't want to do that, but rather we want to get you excited about neuroscience. So we're going to talk a lot about research. We're going to talk about, for example, how neuroscientists are treating depression. For example, how we can measure electricity within your brain and make that useful. How are drugs being developed? So really, we're going to give you a lot of like multimedia stuff, a lot of videos, and you will see, what is OCD like, what is bipolar disorder look like. So my name is Adrian. I'm a college senior at WashU. I study cognitive neuroscience, so really about how you think, as well as chemistry. I am pre-med. I'm really interested in psychiatry, and I've been doing this program for 3.5 years now. I'm here joined by Sophia and Hari. So would you like to introduce yourselves? Sophia?
Yeah, sure. Hi guys, I'm Sophia. I'm a senior also at WashU. I am studying neuroscience and French. I've been with the Brain Bee I volunteered for like 2 to 3 years now. So yeah, it's nice to meet you guys.
Awesome. And Hari?
Hi, hey guys, I'm Hari. I'm a first year. I'm studying philosophy, neuroscience, psychology, a major at WashU, and this is my first time doing the Brain Bee, so I'm really excited to learn and see what this is all about.
Awesome. So the three of us will really try to provide you with a well-rounded experience and get you excited. So with that, just sit back and relax. You don't necessarily have to take notes because we're providing the video as well as the slides at a later date. So really just, follow us and see what are some exciting developments in neuroscience. Okay, so we'll start with chapter 1. So the chapters are in the Brain Facts book, so you can download that book later to make references. But first, we're going to talk about some basics in the brain, anatomy, structure, how do cells look like. So I believe Harry will take over to talk about the brain.
Yeah, so we have a video to show you quickly.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the lobes of the brain and some major landmarks of the brain surface that can be seen from a lateral view. The brain is made up of two halves, known as cerebral hemispheres. When looking at the brain from a lateral view, which just means looking at it from the side, we will only see one cerebral hemisphere, but the landmarks I will point out are found on both cerebral hemispheres. The surface of the cerebral hemispheres is covered with a thick layer of brain tissue known as the cerebral cortex. The cerebral cortex is folded to create more surface area, forming these ridges, known as gyri, and grooves, known as sulci. This large sulcus is known as the central sulcus. It divides the frontal lobe from the parietal lobe. This sulcus is known as the Sylvian fissure, and it separates the temporal lobe from the rest of the cerebral hemisphere. The occipital lobe is found at the back of the brain, but it doesn't have such a clear division separating it from the rest of the brain. There are certain functions associated with the lobes, but any short description of these functions is oversimplified, as each is involved in many different processes. The frontal lobes are important for movement and higher order cognition like rational thought, decision making, and planning. This area in front of the central sulcus is known as the precentral gyrus, and it is essential for motor control as it is the location of the primary motor cortex. The parietal lobes are important for processing of sensory information and are involved in attention and our representation of the space around us. This area in the parietal lobe, the postcentral gyrus, contains the primary somatosensory cortex and is where the sense of touch is processed. The temporal lobe is involved in processing auditory signals and also is important in aspects of learning and memory. The occipital lobes contain major visual processing areas of the brain, like the primary visual cortex.
Okay.
Okay, so that definitely was a lot of information, and if you didn't catch it in the first time, don't worry, I'm going to be going over it again. But if you could go to the next slide, Adrian.
Well,
So yeah, just a quick overview of like what the brain is, and it's like it's a hub of neurons, obviously, and it allows us to think. And inside the brain, there's a ton of like nervous clusters of these neurons. They're kind of like cities. Um, is a good analogy to think about. And as the video mentioned, the brain has this, the surface of the brain, it's like a thin like layer of a bunch of neural cells. I believe it contains 20 billion out of like the more than 100 billion neurons in the brain, is the cerebral cortex. And that's like the outer layer of the brain. And so if you think about it, like if it's confusing to visualize, The outer layer is kind of like how land and water are like at the surface of the earth, and inside the earth there's like, more layers like the crust, the mantle, etc. And that's kind of like how the cerebral cortex is to the brain. So as the video mentioned, there's different parts of the cerebral cortex, which we can see in these pictures. We have the frontal lobe, temporal lobe, parietal lobe, and occipital lobe. So all the lobes. And the frontal lobe, which would be at the front of the brain or like right behind your forehead, that's the lobe that, for the most part, specializes in motor control. There's a portion that really focuses on in the frontal lobe that specializes on motor control. That means that our voluntary, like thoughtful movements, are controlled basically in that brain, a part of the brain. Just below that, we have the temporal lobe, and the temporal lobe, which is like at the sides right above your ears. That's a key point where it is right above your ears. Its mostly functions include processing sound, and it also includes like encoding memories into the brains, and it's responsible for our emotions. But for sound, it makes a lot of sense that it's processed right in the temporal lobe because it's right above your ears. So when you're receiving sounds through your ears, the auditory nerves are going straight to your temporal lobe. The parietal lobe, which is behind the frontal lobe, it's near like the top and like the sides of your brain, is a sensory area that's responsible for the senses that we feel. And by senses, I mean like when you're consciously touching something, maybe like feelings of pressure, pain, temperature, vibrations, movements, etc. So all those senses are concentrated right there. Um, and then we have the occipital lobe, which is like at the back of your head, and that's what specializes in vision. So you can see color, form, shape, and motion. All those neurons are in that area of the brain. So those are parts of the cerebral cortex, but the brain isn't just the cerebral cortex. There's also kind of like an older part of the brain, which is called like the brain stem and like the area around the brain stem. The brain stem's kind of like the core of the brain, or like the old. When I mean oldest, is like evolutionarily oldest. So like the first humans relied on the brain stems more than we do, or not as much. They didn't really have much cerebral cortexes and like that much formation as we do now. They more focused on the brainstem, and so that area is close to where the spinal cord is and the cerebellum. That's just that concentrated area. If you think about like the word stem, it's kind of like in a flower. There's like a stem and there's leaves and petals that come off of a stem, and that's like kind of like the root. And so this is in a similar idea, like the root of the brain. Um, so the parts in this, we have the medulla, so that's kind of like the base area, and it controls like heartbeats and breathing. In addition, with the pons, it works with that part of the brain. We have the cerebellum, and that's kind of a nickname for it's like the little brain, and it's at the rear of the brain stem, and its functions include processing basic senses, coordinating movement and balance, and allowing like you to have nonverbal learning and just memory, remembering things unconsciously. And then the spinal cord, obviously, which is a part of the central nervous system and works with reflexes, and it brings information up into the brain for you to have that info in your brain. And then another key part in your brain is I believe if you go to the next slide, Adrian, is the pituitary gland. It's this tiny pea-sized structure in the brain's core, and it's actually a part of the endocrine system. And the endocrine system is separate from the nervous system, but works closely. And the endocrine system is about hormones. It's all throughout your body. If you hear the word hormones, you might think about growth spurts or puberty. And this pituitary gland, its like main function is to release the growth hormones. So when you start going through puberty, or when you're starting to get really hungry or growing tall, pituitary glands are responsible for releasing the hormones that activates that in your body, and it tells other glands to release hormones throughout the body. And then another part of the brain which is the corpus callosum, which is like connecting the um, uh, left and right hemispheres of the brain. I believe the next slide might show a better picture of this, Adrian. Yeah, so this is a picture of the brain if you slice it vertical, um, and like the half in the middle would be the corpus callosum, the body of it's right there, and it's connected by nerves, but specifically like the axon bundles, and I believe Sophia will get more into that right now. So
Right. So we have a quick question. Does the temporal lobe also carry some visual processing? Harry, do you have comments?
yes, so um, I don't think temporal lobe specializes in visual processing. I think that's mainly the occipital lobe. But one thing I should mention is that the brain is a very complex structure. And it's really important to know that even though these lobes specialize in some things, they also could be working along with other things. I think the brain, um, no one structure completely controls a single function in the brain. The temporal lobe could work in part with vision, if you might have heard that before, but it's primarily the temporal lobe functions with processing sound, encoding memories, and like your emotions, and those are the main functions of it.
Yeah, awesome. Yeah, I remember when I was in class, a good way to remember where temporal lobe is just next to your temples. So, close to your ears. But also on this picture, we wanted to show you, you don't have to memorize all of these terms, but there are many different ways for you to look into the brain. And these different angles provide different resolution to see some of the structures. So if you're just looking at the outer space of the brain, like here, you cannot see the corpus callosum because it's really connecting the two hemispheres. But over here, you can see that very well. It's like a bridge over here. Okay. Sophia.
So now we're going to talk about neurons and like the structure of neuron. Do you guys want to give me a thumbs up if you like know a little bit about this? You can also type in the chat if you want. Okay. So we've got some people who know like a little bit about the neuron structure or the structure of neurons, which is great. So we're going to show you a video. Just pay attention to the structure of the neuron. This might be a review, and then think about how the branching allows it to connect and communicate with other neurons.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the neuron. This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA, or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with the receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
Nice. So just to reemphasize what he said, like these neurons have long arms. They're the axons that can reach to like neurons that are far away. We'll go into this a little bit later too. And then there are also lots of places on these neurons called dendrites where other neurons can come and like dock on and like communicate to the neuron that we're talking about. So like having many flowers on the same plant, you need multiple places where other like bees for flowers, but like neurons can communicate to this neuron in order for it to communicate to others. So if we go on to the next slide, this is just another overview of the neuron. So the cell body, as he said, contains a lot of like the organelles, nucleus, your mitochondria are there, and it also has the dendrites, which are the places where the neuron takes in information, and then when it wants to send information out, it goes it sends an electrical signal down this long appendage called the axon, which can have myelin sheaths on top of it, depending on whether it needs to send signals long distance. And we'll go into the nodes of Ranvier in the next slide, I believe. And then there are these axon terminals on the very end of the axon that synapse onto other cells. So pretty much just like they approach other cells and they communicate to them through there. Yeah, and if you have any questions, feel free to post them in the chat, or you can like raise a hand or something. All right, if we go to the next slide, here's just a picture of what neurons look like in real life through microscopes. So on the left, we've got, this is called crescent violet staining. So you can see the cell body. It mainly stains the cell body, but cell body, the nucleus, and the nucleolus, which is inside the nucleus. And then on the right, though, this is an image looking down the axon. So if you think about the axon being that hollow tube, we're looking through it, which is why you have that myelin sheath on the outside, which is that dark black matter. And then a Schwann cell is just a cell that like makes and maintains the myelin sheath. Yeah. On the next slide, so neurons communicate to one another through these electrical signals. So when a neuron wants to send a signal, it'll send that electrical signal down the axon, as I mentioned before, to the synaptic terminal. At the synaptic terminal, it triggers a release of chemicals called neurotransmitters that like physically travel across this little gap between cells, between one cell's axon terminals to another's dendrites. So they'll diffuse through the synapse, these little chemicals, and then bind to the receiving neurons, which can either turn up or turn down. So it can tell the following cell to either stop or like go. It can turn up or turn down the electrical activity in the receiving neuron.
Yeah, if I may just add one more thing. So, so just remember that neurons usually use electricity. So one of the really cool things about that doctors can do is we can measure the electricity in your brain, right? Because all of your neurons are communicating using electrical signals. So we'll talk about this later, but scientists have developed this really cool equipment that basically just goes onto your head and it measures electrical signal in your brain. And that is because neurons communicate using electrical signals.
Yeah, that one's a surface level one in this picture here, but later on we'll see one that like actually gets placed into the brain. So yeah, no. The neuron sends, so the electric signal travels down its own axon, and then it sends neurotransmitters through, so it doesn't actually send the electrical signal through the from one cell to another.
Yeah. Okay. So on this Sophia, are you seeing this myelination slide?
Yes.
Okay, perfect. So about myelination, oh, there's someone in the, okay, perfect. So in this myelination process, remember that axons, or this long branch from the neurons, they carry electrical signal. But think about what in real life carries electrical signal. Wires, right? And wires, we don't really just see the bare wire. We will see that that's, covered and surrounded by this layer of plastic or rubber, right? So we call that insulation. Neurons have a similar process, and they call that myelination. That the axons are wrapped in myelin, as Sophia just told you. But why is that important? Well, because myelination is a form of insulation that would preserve the strength of the electrical signal passing down the axon. And because of this wrapping, electrical signal can travel way faster. So here you're seeing, if there is myelination, then electrical signal is this fast. But if there's D or hypomyelination, meaning there's a lack of myelin, it travels way slower. And a lot of times we do want electrical signal to travel fast because there are a lot of instantaneous stuff that you're doing, right? And you certainly don't want, whatever thought process you have or whatever reflex you're doing to take several minutes because that is just damaging. So because of myelin, a lot of the axons are wrapped and insulated, but there are little areas that are not insulated, and those are called the nodes of Ranvier. And because of that, action potential, or you can think about electrical signal, really just jumps at these nodes of Ranvier. They travel very fast through the axon because they're jumping through a lot of the length of the axon. So that is called saltatory conduction. Okay. And then I also wanted to show you some really cool things that doctors are currently doing. So in this video, people are talking about depression, but people are showing how, stimulating a part of your brain or stimulating multiple parts of your brain using electricity can really change how the brain functions. And remember, that is because neurons communicate using electrical signal. Okay, yeah, so in this video, what you saw is what we call a neural circuit. So scientists planted these electrodes into this person's brain. And the powerful thing is that, this person has really treatment-resistant depression. Now, in the stages of depression, there is abnormal brain activity going on. So these electrodes will then signal and say, hey, there's something going wrong in this electrical activity. And what I'm going to do is I'm going to signal I want to stimulate this area so that it can go back to normal. And that's all done through what we call deep brain stimulation. So just really think about how cool this is. You're really transforming what you know about the brain into really helping other people. And we're talking about really treatment-resistant depression, and this has really became, a really, really cornerstone in tomorrow's psychiatry. Okay, and now we're going to have some practice questions. These are just really short and sweet. I encourage you to, to unmute yourselves and to shout out the answers with us. So the first question we have is, which of the following are part of the central nervous system? Anyone have a answer? Okay, I see a lot of you are muted, so just okay. Oh, you don't have to raise your hand. Just so I see Rebecca.
Is it spinal cord and brain?
Spinal cord and brain. Perfect. So remember, so of course, the brain is part of the central nervous system, but also remember you have the spinal cord, right? And they're connected at the brain stem. And you don't have to raise your hand or anything. A and D. Okay, perfect. So great. Let's move on to this question. And right, you can just shout out your answers. So after a stroke, which stroke damages part of the brain, a patient presents to you with loss of visual perception. So their vision is not going all right in an area of the visual field. But interestingly, their eyes are completely normal. So what part of their brain is more likely to be impacted? So I have the four lobes here: frontal, occipital, parietal, and temporal. Does anyone have a guess?
Would it be parietal?
Right. Well, yes, but parietal is more involved in, touching, where it's, we call that somatosensation. It's about like you feeling onto something.
Could it be the frontal lobe?
Well, frontal lobe is certainly involved in a bit of a processing, but frontal lobe is more about, emotion, motor sensation, well, motor activities, how you move. And we also call that executive functioning, controlling yourself, regulation. So I see someone in the chat put, okay, right, Savani and Michael, occipital lobe. So the answer is occipital lobe because, it's in the back of the brain, and this is actually the first place that is, involved in higher processing of vision in your brain. So the answer would be occipital lobe. I know it's a little bit counterintuitive because it's literally, the farthest from your eye, but there's a part of this lobe we call V1. So that is primary visual area. So there's a bonus discussion which we didn't really touch on. So if this patient has vision loss in the right field, which side of the brain is likely to be affected? It's either left or right. Okay, so I see Savani saying left. Yes. So there is this crossing mechanism in your brain. So the visual information that your eyes receive gets passed on to the brain. But at this location, which is called optic chiasm, it looks like an X. So information from your right field travels to the left part of the brain. And information from the left field travels to the right part of the brain. So remember, there's this crossing mechanism, and this really exists in a lot of senses. So it's not just to vision. All right. So I have a follow-up question to that. So remember, there's this crossing in your brain going on, right? So you have to really think about left versus right. If an individual is only able to comprehend sounds that come in through the right ear, which part of the brain would they have damage to? Anyone want to shout out their answer?
Would it be left temporal lobe?
Left temporal lobe it is. So remember, temporal lobe process, sounds, comprehension, and because we have this crossing going on, you have to go to the left. And perfect, Savannah, perfect. Okay, I think this is the last practice question for this. The part of a healthy neuron that speeds up electrical signal transduction. So would that be axon, synapse, myelin sheath, or nodes of Ranvier?
Would that be C, myelin sheath?
Perfect, yes. The answer will be myelin sheath. Remember this insulating layer. So a bonus discussion we didn't talk about is what can damage this structure. So this is actually some part of my research. So when people have inflammation going on in their brain, their myelin can, be interfered with a lot of like immune cells. And these immune cells can actually damage myelin. So we call that demyelination. That is like a removal of healthy myelin. And unfortunately, in people with inflammation, we do see a lot of impairment in terms of thinking, cognition, or sometimes even motion. So yeah. All right, so next we're going to move on to chapter 6 to 8, which talk about brain development. So it really starts when there's simply an embryo, sort of fertilized egg, and it goes all the way from childhood to adulthood and then to geriatric or the elderly. So how does the brain change over time? Sophia?
Yeah, so again, as Adrian said, we all start as like a single cell, which is kind of crazy. So this video is going to kind of talk us through how the brain develops. Just pay attention to, there's a lot of terms. Pay attention to the ectoderm, the neurotube, and then development of encephalon structures. Encephalon just means brain, so the different brain structures.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss early neural development. The development of the nervous system begins at around the 3rd week of embryonic development, when an area of the ectoderm, or the outer tissue layer of the embryo, thickens and forms what is known as the neural plate. This plate begins to fold inward, forming a groove called the neural groove. The sides of the neural groove, known as the neural folds, begin to come together. At the end of the 3rd week, the folds will begin to fuse together. By the end of the 4th week, they have completely fused together to form the neural tube, which will eventually become the brain and the spinal cord. As the neural tube closes, bulges and bends begin to appear and they gradually become more noticeable. During the 4th week, there are 3 of these bulges present. They are called the primary vesicles. They are the prosencephalon, which will eventually form the cerebrum, the mesencephalon, which will eventually become the midbrain, and the rhombencephalon, which will eventually become the rest of the brainstem and the cerebellum. The end of the neural tube will form the spinal cord. As the brain continues to develop, 2 of these vesicles further subdivide to form secondary vesicles. The prosencephalon forms the telencephalon and the diencephalon. The telencephalon will become the cerebral hemispheres. The diencephalon will eventually consist of the thalamus, hypothalamus, and other structures. The mesencephalon does not subdivide any further and will become the midbrain. The rhombencephalon will subdivide into the metencephalon and the myelencephalon. The metencephalon will become the pons and the cerebellum, and the myencephalon will become the medulla. The spinal cord will still form from the end of the neural tube. Next, the neural tube will continue to develop to look more like the brain. The telencephalon will grow more rapidly than other parts of the tube, and by 11 weeks, the brain will have a similar shape as to what it will at birth. Although it will continue to develop after birth, the brain at birth is structurally similar to a fully developed brain.
Okay, so let's the embryonic brain development stages that we're going to go through are induction, neuronal migration, neuroconnection and myelination, and then pairing back to the neural network. So a lot more simpler than what he just went through. But if we go to the next slide, let's start off with induction. There's 3 different cell layers that are formed super early on in embryonic development. There's the endoderm, which is on the very inside, mesoderm, and then ectoderm. So a good way to remember like which one's which, if you look at the prefixes, so endoderm, endogenous, endo, like means inside. Meso, if you think about like, the only example I can think of was Mesopotamia, but like that's between 2 rivers, it kind of means middle, and then ectoderm is outside. I don't have a good one for that one. So these cell layers get messages from nearby tissue, which then tell them what kind of cell types to become. So the ectoderm is the outside layer, and that's the part that we'll talk about mainly because it forms parts of the body like the skin, the hair, but most importantly, the nervous system. And this process of neuroinduction is when the ectoderm cells get the green light to turn into neurostem cells, and then these stem cells then differentiate and turn into like neurons or glia or other types of like brain cells and become more and more specialized. If we go to the next slide, there's this really, really cool gene called the sonic hedgehog gene. It's a signaling molecule. The gene produces a signaling molecule, this protein, and that is actually how cells, it determines what kind of cell is being formed. So if we look at the little diagram here, the blue, light blue, are sonic hedgehog producing cells. The cells that are near the release of these sonic hedgehog producing cells become glia cells, which are helper cells. They're more not auxiliary, but they help the neurons. Cells that are produced farther away from the sonic hedgehog producing cells become motor neurons, which are the neurons that control your muscles, etc. So what kind of cell it forms is affected by how much of this sonic molecule is actually there, which that was pretty cool. If we go to the next slide.
Oh, sorry. If I would just re-emphasize the key takeaway. So remember, everyone, the ectoderm goes into forming your nervous system, but the ectoderm doesn't just do it by itself, right? It receives directions and guidance from the mesoderm, and part of the mesoderm that Sophia just talked about would be this sonic hedgehog gene. So how much of this molecule there is really influences what kind of neuron or what kind of cell the ectoderm is going to turn into. Okay.
So after induction, technically there's a step of proliferation where lots of multiplication happens. And a lot of neurodevelopment problems can happen for the baby during that time. But after that, you have migration. So once induction occurs, the neurons have to go to their final destinations, from the ectoderm to their final destinations. So 3 to 4 weeks after the baby's conception, these cells, while they're dividing, they form a flat neural plate, as you saw in the video, and then becomes a neural groove. And you get these parallel ridges, and it fuses eventually to form a hollow neural tube. So you can see the tube kind of on the left there at 3 weeks. It forms, so the little head part, I know it only says forebrain, but the head part of the tube is supposed to form 3 bulges. It has a hindbrain. It forms the hindbrain, the midbrain, and the forebrain. And you can see the spinal cord in the middle there. And then there's also different guiding cues that make sure the neuron gets to where it needs to be. So about 90 to, I think that's the next slide actually. Nice. So so new neurons are guided to their destination by glia, these glial cells, and they act as scaffolding in a way. They help move the neurons to where they need to be. 90% of migration actually in your brain involves radioglia, which is a specific type of glia. And during this stage of migration, cells can be really heavily influenced. So they're kind of vulnerable. So alcohol, drugs, radiation that are in the mother's environment can affect how the baby develops during this stage. So that can result in epilepsy, can result in disability. There's actually a disorder. It's a disorder that's can be fixed now, but it's called spina bifida. If you look at the right there, there's a picture of this baby, and it's got the spinal cord that's, instead of being inside the vertebrae as you expect it to be, it's kind of jutted out. So that can result, I think, from improper migration during this period of development. Does anyone have any questions? Feel free to post them in the chat. Okay, well, you can keep posting. So once the neurons have gotten to their final destination, they can start forming connections with one another. They're setting up those communication kind of endpoints. And as we mentioned before, the axons and dendrites send and receive signals respectively between neurons. So they start, the axons start lengthening during when they're starting to, when the neurons start trying to make connections. So there are these little growth cones, which are on the axon's tips, that seek out a precise destination using molecular paths in the environment to find like where they're supposed to go, like the neural connections they're supposed to make with like specific neurons. And then once they've gotten to their destination, the axons form these little like gaps where they form these synapses, which is like a special connectional gap where the neurotransmitters go across. So in the video here, we can actually literally see synapses forming and like axons lengthening and stuff.
Yeah, so in this video, you see, there are these neurons, there are cell body, but you can see that these different neurons are kind of like branching out and they're reaching out for each other. So this is what we meant by making connections. A lot of your brain functions, especially those complicated ones like thinking, decision making, has to do with extensive connections between many, many different neurons. So it's like a whole network.
Yeah, so once the neurons have formed all these different connections, you've got a very complicated neural network, which is important. Your brain is complicated, but some of these connections aren't necessary. So now there's a stage of paring back, where you remove the incorrect connections and identify the correct ones. So actually about half of the neurons created in development survive until adulthood. That might be partially due to death, but also partially due to the fact that we like pair off or pair back the neurons. So the neurons that are unnecessary go through apoptosis, which is a programmed cell death. So it's built into their program, kind of. So if a cell isn't receiving enough of these life-sustaining chemicals called neurotrophic factors, the cell dies. Actually, this is a quick plug for WashU. Nerve growth factor was actually discovered by a WashU professor. She got the Nobel Prize for it, which is kind of amazing. And she's below there. Rita Levi Montalcini, I think. So the neural network is thus like cut down so that it's complicated enough, but also doesn't have a bunch of incorrect or unnecessary connections. Just like how bushes need to be pruned, we need to prune the excess branches off of our neural network during a stage of development. Okay. All right, so we've mainly covered what neurons are programmed to do before birth, but the brain continues developing until you're in your 20s. I think it's like 25 when your frontal lobe is finally developed. So what about a newborn or a child's environment? It's important that kids have certain sensory or movement, emotional input, like these experiences in order for your brain to like take in and continue developing. And plasticity is actually the word for the ability to adapt to these environmental factors. So studies have shown that there is actually like a critical period, a time before or during which you need to have certain experiences in order to develop healthily. So for example, there's a critical period for developing language. So your ability to learn language, I'm sure you've heard this, diminishes over time. So teaching like a young child before 3 to 7, apparently, or before 8 years old, they'll learn best. They'll learn the language most fluently, like if you teach them before that time. Yeah, so there's also cases of children who haven't been exposed to like literally any language at all, and until like elementary or middle school, and they have a hard time learning even like a first language. And so because they haven't had that experience early on, they can't develop those skills. There's also a critical period, I think that's kind of related to tone development. Children with better children can develop better musical abilities, like absolute pitch, perfect pitch, than adults. But then this critical period is also important in that the brain is also very sensitive. So if you have abnormal brain activity, that can also lead to a lot of childhood disorders like epilepsy. So all in all, it's important. There's like a very early period of time when you have to have those experiences or inputs in order for your brain to develop healthily. And these are two cool cases of just to show you how amazing the brain's plasticity is. On the left here, this is a kid, this is a study about this one kid who had, he had bilateral strokes. So that means he had strokes on both sides of his brain 3 weeks after birth, which if you had that in an adult, it would probably result in death or severe like mental deficits. Because when you have a stroke, parts of your brain die off because they don't have the nutrients, the blood, the oxygen they need. So if you look at the picture, the black spaces in A are where he doesn't have brain. Like those are just spaces. But because he had it 3 weeks after birth, his brain was still developing, so it adapted. So they didn't actually know. He developed perfectly fine. He only had like slight motor deficits, and they didn't know until he was like 13 when he had some clumsiness in his right hand when playing baseball. So they brought him into a neurology clinic for a scan. Turned out he has an abnormal brain. His chunks kind of missing, but he's developed perfectly well, which is a kind of just speaks to the plasticity of your brain. And then on the right here, it's called the pink cast study. This was a study done on, I believe, adults, where they put a cast on the subject's like dominant arm for 2 weeks, and they were looking at like brain activity, and showed that parts of the cortex and the cerebellum, as Hari was talking about earlier, those are like parts of those two regions controlling the arm were like functionally disconnected while the cast was on, because movement was so limited. And then after cast removal, the activity like returned to normal. So yeah, that was just another cool study that shows how the brain kind of adapts depending on the body, depending on environmental factors. Yeah. All right, so we can move on to practice questions. Okay, so the first one is, which layer of the embryo gives rise to the nervous system, the brain? Your choices are ectoderm, mesoderm, endoderm, and the nervous system is not from the embryo. Feel free to unmute or post in chat or raise a hand.
I think this might be a challenging one. So Sophia, would you just remind us on the answer?
Okay, we've got to vote for C. That is the endoderm. It's actually responsible for more of like internal organs, if I'm correct. So the layer of the embryo that gives rise.
The ectoderm?
Yes, it is the ectoderm. Ectoderm gives rise to the nervous system. So ecto, outside. I know it's a little counterintuitive because you think about the brain or like the nervous system being inside, but it's the ectoderm. So bonus discussion question, which layer induces nervous system genesis and differentiation?
Is that asking like which layer is like responsible for making like different types of neurons?
Yes.
Is it the mesoderm?
Yes.
Okay.
Nice. All right. Practice question 2. Okay, how do cells in the ectoderm differentiate into specialized neurons? A, through guidance of signaling proteins like sonic hedgehog. B, through traveling along radioglia scaffoldings. C, through signaling from nerve growth factors. Or D, through apoptosis. A? A is correct. So sonic hedgehog, again, determines what kind of cells depending on how much of the sonic hedgehog is like present.
Perfect. So, and then thank you, Sophia. We'll now be moving on, how we can study the brain, how we're actually looking into the brain, right? Because when we are talking about, specimen or animals, sure, we can do dissections. But when you go into a clinic and you complain, hey, I seem to have a headache or there's something going on, you definitely don't want your brain to be taken out. So how do we look into the brain while it's still within your body? I think Hari will lead this section.
Yep. So there are two ways that neuroscientists study the brain. It would be studying non-human animals and studying humans. So first of all, animal research. There's plenty of research done on animals. Mice and rats are actually the most common research in general, like neuroscience, when people are just studying the brain in general, because rats have like really similar brain structures to humans, and it's really easy to relate the findings in them to us. Slugs are useful for researching memory. We use zebrafish to understand like neurological processes. Pigeons are good for like color vision and visual memory, and just each different animal has like their own upside, and just studying each of them could be useful for us in different ways. Um, there's some cool examples in the bottom. For like squid, for example, we can understand ionic interactions in the action potential. So action potentials within the axons and the nerves. We can understand how the ions there or the chemicals move throughout those axons just by studying through squid, because it's a lot easier to study in squid. Same thing goes. There's some things for chicken and frog, and then etc.
Yeah.
And so
So someone talked about fruit flies and mice. So both of them are very commonly used. Fruit flies are more involved in genetic studies because they just, you can have a lot of generations very quickly and they develop, different appearances. Um, okay.
yes, and then so those are ways we study animals to understand more about our human brain, and we also can study human brains themselves, and it's in a more complicated process. So, as Adrian was mentioning earlier, neurons communicate through chemicals, right? So we're able to interpret those electric signals or chemicals through understanding electrophysiology. And electrophysiology basically means just studying those electrical properties. And there are plenty of ways that we can measure them. For example, the one mentioned on the slide, electroencephalography, is a really common method that people or neuroscientists use. And it's probably what you're used to seeing in especially like sci-fi movies, when people put like a bunch of nodes around the brain, and what they're trying to do is to record the waves of electrical activity that's going on inside the brain's surface. Another way it's also used is in hospitals, and it helps doctors find brain disorders or seizures, and EEGs. You can see like the readings on the right where they have like the spikes and going on, and if there was like, for example, something wrong going on in the brain, such as like a stroke or a seizure, there'd be like those rapid spikes that would be happening really quickly. So there's a lot of use for electroencephalography. Next slide, please. And then another form is positron emission tomography. So what is used in this form of measuring brain activity is that they radioactively tag like a glucose molecule. So I believe it's usually carbon. They tag like carbon 14, which is like a less common carbon that you don't normally find. And so by tagging it, they're able to measure where it goes throughout the brain, and they know that what happens or goes around. They can track where it goes around, and then they can basically understand brain structures. and like relate these structures to different states of mind. So, for example, on the left side, um, we can see that we can track those uh, where the radioactive tracer is going, and then we can understand how the brain looks in a vegetative state, or maybe a less conscious, or like a fully conscious state. It's also important to see, say, you want to compare two individuals. Maybe one has like no neurodegenerative diseases or just no like disorders, and then another person might have like a dementia or like Parkinson's disease, as seen in the right picture. We can see that their brains are definitely different, and um, there's something else going on within the brain that kind of gives scientists the certainty that they can say, oh, that someone with Parkinson's disease does have a brain that's different from someone with a normal, and then they can investigate further and try and understand where it's coming from. Also, to quickly recap how PET works, is that there's the tracer which goes through the blood, and then because it travels when there's neural activity, you need blood flow that goes through that. So that's how we're able to track the blood flow, because there's something called a positron, which is related to like electricity that we can track. Next slide, please. And then MRI, you might have heard this one before, definitely used in hospitals a lot. It's a really common form of basically just um, measuring high quality images of basically any tissue, but it also is really useful for the brain because the brain is such a complex organism that has a lot of structures packed tightly into it. So it's very useful to get high quality images of the structure and for scientists to basically differentiate different parts of the brain. So how the way it works is that they use a magnetic field. You can kind of see on the left side where they put someone in a big tube, and it's like kind of scary. But it's really all they're doing is that they're putting someone in magnetic fields, and they use radio waves to basically interact with the atoms in the soft tissue that they're analyzing. So, say the brain, they would be interacting with the atoms in the brain, and it'd be and the magnetic fields, and it would basically help draw a picture of like the waves that are given off from the brain, and that helps us really see here, the parts of the brain that there's stuff there, or parts that it's dark, which means that there's nothing emitting electricity. So bones usually, which are solid, they won't really emit electricity. So that helps us understand where the bones are in our brain versus where like organs which have atoms that will emit electricity are. I believe there's a question in the chat. On the PET, what do the different colors represent?
Okay,
So if you
so
can go.
oh, if I might just take this quickly. So the really charm of PET imaging is that you can really radioactively tag anything that you want. So how it does talk about, it can be glucose, which is normally what your brain would consume, but also in other conditions like Parkinson's, there is this known neurotransmitter called dopamine that is deficient in people with Parkinson's disease. So if you tag that, what you see is that people with Parkinson's in this brain area called the basal ganglia, they don't have a lot of that, right? So brighter colors or more reddish colors means higher concentration. Where you see in normal people's brain, they have a lot of dopamine in this area. So really the colors mean relative concentration. Okay.
Yes, and so that is for PET, and then also MRI, and then we have functional MRI, which is a little slightly different from MRI because it allows to show neuronal activity with like a high structure of the brain, and then we can kind of compare their activity under resting and active conditions, and you can see in this image. Oh, sorry, hold on one second. So there's like you can basically understand the brain at different stages. It's similar to I think what was used in PET, where you're understanding um, the brains at different stages. But you can also it's a little bit of a combination because a lot of these uh, measuring strategies are kind of you can use many different from this for the same type of purposes. This is also MRI where you could understand the spatial resolution, and it combines that to help mainly detect like the blood oxygen levels, and so that helps understand the brain and how it uses oxygen and where the blood levels are going throughout the brain.
Right. So just to complement what Harry was saying, when you are doing different tasks, let's say you're thinking or you're speaking with someone, this certain parts of your brain will start to use more blood. And the reason of that is because it needs more oxygen. So this is what this graph is showing. Let's say this is your thinking process. When you start to think, your brain starts to require oxygen. So you would have a shoot up of oxygen, but then interestingly, oxygen consumption or the use of oxygen is rather slow. So what you would have is you have a really huge buildup of oxygen at certain places in your brain. So what we're measuring using functional imaging is we're measuring this relative concentration. So this work is pioneered here at WashU, but really what you see here is let's say you're hearing words, you see a lot of oxygen buildup in your temporal lobe, right? Remember, temporal lobe involved in hearing and involved in sound comprehension. If you're speaking, there's this motor area. If you're thinking, it's your frontal lobe. If you're seeing, it's your occipital lobe. So functional imaging is an awesome way for us to visualize the different parts of your brain that are being activated. And of course, it always needs a contrast, right? So we always contrast it to, let's say you're not doing anything, so that I can really see, oh, if you're just seeing words versus you're not doing anything, this part of the brain that gets activated is probably involved in seeing words. Okay, so I believe, Howard, do you want to take over this?
Yeah, I think all I had was just to show the video. I think it would,
Okay.
yeah.
After years spent imaging brains, Marcus eventually discovered an entire mental network that coordinates our movements with our senses. And it turns on the moment you stop thinking.
Nobody was even looking for this. It was almost by accident. It came to our attention that if you just were laying in a scanner and we were looking at your brain and then we asked you to do something, not only did things go up, but certain things went down.
Certain parts of the brain seem to turn off whenever we begin a task. Those same regions become highly active whenever we are quiet and relaxed. Marcus calls these linked sections of the brain the default mode network, because the brain defaults to this activity whenever we are not doing anything else. In fact, the brain is just as active in this default mode as when we're consciously doing something. After careful analysis, Marcus thinks he understands why.
The default mode is deeply important in creating an ability to predict what's going to happen next. I think it's really critical.
In the same way that an ensemble's musical director organizes the group's music, the default mode network organizes all the functions of the brain so that mind and body are always on the same page. But is our subconscious always the servant of our conscious mind? Or can it also take charge? The subconscious is constantly at work behind the scenes. Without it, we'd never make it through the day. But could that hidden brain power do even more? Some scientists believe we can tap into our subconscious to heal our bodies and expand our minds.
Okay, so the key takeaway from here is remember functional imaging that you just saw is like measuring oxygen buildup when you're doing something. And different parts of the brain will get activated differentially when you're doing different tasks. Now, there are parts in the brain that would always co-activate together. So for example, in the visual area, all this blob activates together when you're seeing something. When you're trying to control yourself or expressing emotion regulation, these different parts, even though they're not actually connected, they show the same activation patterns together. And interestingly, here at WashU, scientists have discovered that there is this network called the default mode network that actually goes down in activity when you are starting to become more active. And it goes up in activity when you're just, daydreaming or resting and you're not doing anything. So just know that there are different networks that co-activate together, and each network is responsible for a different activity. Okay. And some quick practice questions. Harry, do you want to lead this part?
Yeah. So, which of these does MRI use to produce neuroimages? It's in the name, by the way.
Is it magnetic fields?
Yep. Magnetic fields. Yeah, magnetic resonance imaging. That is what MRI stands for. So yeah, it uses magnetic fields in that big machine right there. And yeah, we have a bonus discussion. What about EEG and fMRI? What do they use? If anyone has a guess, go at it.
Would EEG use electric fields?
I believe that is right, if I'm right,
Yes, that
Adrian.
is correct. So electroencephalography that measures electrical activity. So that's more about measuring electricity, which would use electric fields. Now with functional MRI, interestingly, it also uses magnetic fields, right? Because you still are using a sort of magnetic resonance imaging. So I put in this picture to remind you that MRI uses magnets. So, and that is why if you've ever done an MRI scan, you will be told you cannot have any metal to bring into the room. Now, this is a accident that happened because someone brought in this chair-like thing into the scan room and it got immediately sucked into the machine, damaging the machine. But imagine like if you have like a metal transplant in your brain, like in your body, that would be super dangerous. So people with metal transplants cannot actually do MRI. They would do some other imaging like EEG or infrared something. Okay.
Alrighty, so what is the best technique to use for the following study? So you're walking in Forest Park and you bumped into your friend, you talk to her, and you feel you're happier than before, and you wonder if your talking is related to some brain activity changes that would explain changes in your mood. And so, what would you be able to? What technique can you use to help understand?
Could you use a PET to like measure like a certain hormone or something that's associated with like happiness?
Yeah, using a PET is a good idea. The problem with PET, and that's why it's not as popular, is because A, remember, you always have to use a radioactive tracer. And for that, you need to use a needle to inject it into your body. So it's more like invasive. The other thing about PET is that it's really slow. A lot of radioactivity has a, what we call a long half-life. Basically, it just takes a long time for it to show any effects. So beyond PET, what would be a good technique to use here? Okay, anyone has a guess? All right, um, so Hari, do you want to explain what the next choice is?
Yeah, I believe it's fMRI,
Uh-huh.
and that's because, as Adrian was just saying, PET is really a slower process, and it's not really that used that often. But fMRI, on the other hand, has a lot of similar, like I said earlier, it could also be used for similar things that PET can be used for, and it can help trace the oxygen flow of throughout your brain and how that helps understand different parts of your brain and when it's being activated at different points in your emotions. So yeah.
Yeah, and then we have a bonus question or discussion for you to think about. So remember, with functional MRI, fMRI, you always need two conditions, right? You always have a one experimental and you have one that is controlled. So in this case, if we're trying to decide, well, is there any brain activity associated with change in mood? What would be the two conditions? So remember, you have control. Okay, Rebecca, I see you having,
Okay.
okay,
Would the control group, will the control like fMRI be where the person isn't talking and then the experimental be where they are talking?
exactly, exactly. Awesome answer. So the control is always, you are at rest. You're not doing anything. And experimental here is, you're doing something where you're engaged in something. And people would, subtract the brain pattern in these two conditions. So you're really looking for the difference, right? And that difference might just explain some of the brain activity associated with this added on experiment you're doing. So everyone, I hope you really had a good time with this tutorial. I know it's a little bit kind of heavy, but really try not to focus on the nitty-gritty. Instead, look into, the experiments, how scientists are using these concepts to try to, understand diseases, understand health, and help other people. We talk about deep brain stimulation, and that is reliant on how your brain uses electricity, right? And eventually how that's being used to help people with depression. We also talked a lot about, imaging, how we can look into your brain and see how it is activating without, really cracking the brain open. So that has allowed us to understand how thinking works, how emotion works. And then you also learn a lot about, development that Sophia talked about. During development, you have this neural tube going on. The neurons will need to go into right places, right? And then after they settle, they need to make connections. And a lot of these connections will need to be shaped by your experience. So a lot of things can go awry in those processes. And that is why, during pregnancy, women are encouraged to take a lot of like nutrients, like vitamins, like folic acids. So that's really to make sure all of these developmental processes that are so vulnerable that can go perfectly normal. And we also talk at the very beginning, remember the different parts of your brain. There are different angles to look into your brain. And then the brain is made up of neurons, right? Neurons have axons that are wrapped in myelin sheath, so electrical signal can go faster and faster. We didn't really talk about developmental and psychiatric conditions, but that is something we'll cover in the next tutorial. So the next tutorial, if you want to, see how a doctor really works and see what patients the doctors would see, really come to the next tutorial because we'll be talking about a lot in psychiatric conditions like autism, like depression, bipolar, schizophrenia. We'll also talk about how thinking works. What is cognition? How do I control my everyday behavior? So just a reminder, so again, the Brain Bee is settled on February 26th. So we do require a registration. That is to keep you in our system. We'll send you all of the news and relevant information. We'll also be sending out neuroscience at home toolkits. So we'll send that either to the school or home. We're still in the process of coordinating that, but just know that there will be this exciting toolbox coming to you to explore a lot of the things that we just talked about. So I do encourage you to register for Brain Bee to participate. You guys are all bright and eager, and you guys really just learn a lot about neuroscience.
Adrian, I'm sorry, if I could cut in real quick before you finish up. For all my yes teams that are on the call, the email was sent to you. Make sure that when Adrian dismisses us, that you register so that everything that he's talking about is in your inbox, and he will send all the information regarding us meeting again next week. Sorry, Adrian.
That is wonderful. Thank you, Ms. Harsley. And then the next tutorial will be meeting in 2 weeks. So we're not meeting next week, but we have another session on the 5th at the same time. It's also a Saturday. So I hope to see you there and talk a lot about diseases, psychiatry, thinking, really more things that are relevant to your life. Okay, so this tutorial is officially over. You can log off this meeting. Thank you so much for coming. And Sophia and Harry, would you just stay for a little bit of time? I just want to recap a little bit. But everyone have a wonderful Saturday. Thank you for your participation.
Don't forget to register, my teams. But we will meet different next week, my yes teams. So be on the email for that. Hey, Adrian,
Yeah.
how soon will you be sending out the link for that video?
I will be sending out that today.
Wonderful. As soon as you send it out, I got something for my teams that weren't there. Thank you so much. Bye, my team.
No problem. Great to see you.
And I want to tell you guys, this took me back to high school. So I remembered a lot of stuff that I did learn in anatomy and physiology. So great job.
Wonderful. Wonderful. Thank you so much for the.
All right. Bye, you guys. Have a great Saturday.
You too.
Bye.
Thank you.
Okay. Any questions?
Stop the recording.
Yes.
Oh,
I'll do that now. I just